My name is Autumn Sylvestri. I'm studying biological engineering, specifically in the bioprocessing concentration in plant biology here at NC State. I'm working as a REEP Scholar under Dr. Kolar, and for my project I'm focusing on valorizing the products of lignin. Plant biomass is composed of three main components, which are cellulose, hemicellulose, and lignin. And while hemicellulose and cellulose have effective processing methods, the two of them can be converted into biofuel and similar products that are helpful and renewable since the source is the biomass itself. But lignin does not have an effective processing method. It's mostly discarded as waste or burned for energy, neither of which are very sustainable. Broadly, as we see the human population increase, we need to shift to more sustainable methods of production. For biomass to compete with the petroleum industry, we have to make it an effective competitor in the sense that nothing is being discarded as waste. I'm looking at lignin and treating it with biological catalysts and chemical catalysts to see how the molecule breaks down, what it breaks down into, characterizing those products, and seeing what valuable end products can be produced through these treatment methods. The catalyst I'm using is actually produced by Nitesh in the lab. Adding the catalyst to the lignin and allowing it to run for 25 minutes, taking samples every 5 minutes to see how the molecule breaks down if it reacts with this catalyst, what products are produced, and in what quantity that they are produced. So I've taken the samples and added them to a solution of methanol and then added them to the gas chromatography mass spectrometer machine, which is going to characterize the samples themselves one by one by determining the relative concentrations and the characterization of each molecule that's in the sample. So on the computer screen, we will see chromatograms, which are peaks drawn out with the relative areas, and the higher the peak, the greater the relative concentration of the product in solution. If the product forms and then further diminishes, we know that it's oxidizing too much, and we have to adjust the experiment to see how we can maximize the yield. Some examples of end products of lignin when it's broken down are, one notable one is vanillin, which is a synthetic flavoring that's used to mimic vanilla in biopesticides and in cosmetics and fragrances, because the molecule itself is very complex and breaks down into phenolic and aromatic compounds. At NC State, and especially in the biological engineering department, there's such a significant focus on making sure you have hands-on experience. Dr. Kolar has been a wonderful research mentor. He's always happy to answer your questions, and he gives you the tools and the space and try to figure out the procedures for yourself.